Have you ever really stopped to think about how an animal is built? I mean, forget about skyscrapers or massive bridges. The most incredible construction project on the planet starts from just a single cell. So today, we're going to look at animal development like it's the ultimate masterclass in architecture, starting with a blueprint that's literally microscopic.
And that right there is one of the biggest mysteries in all of biology, isn't it? How does that one single cell, this simple little sphere, somehow contain all the instructions to build the insane complexity of a living, breathing creature? That's the huge question we're going to try and unravel.
You know, every big construction job needs a blueprint. Before you can even think about putting up walls or deciding where the kitchen goes, you have to know which way is up, down, left, and right. Well, in biology, the very first problem is exactly the same. How do you establish a basic body plan from what is, essentially, a formless blob?
So this first critical step is called axis formation. Just think of it like drawing the X, Y, and Z axes on a piece of graph paper. It creates this fundamental coordinate system for the embryo, and it tells every single cell that comes after where it is. Is it near the future head, the tail, the back, or the belly? Without this, you just have chaos. And honestly, when this step goes wrong, it's a primary cause of major birth defects, which just shows you how crucial it is to get that blueprint right from moment one.
So, how in the world does this even happen? Well, let's take a look at the humble fruit fly. In this case, the mother basically preloads the blueprint into the egg. Before it's even fertilized, she packs it full of these chemical instructions, these little messenger molecules that are going to guide all the first big decisions.
And the way it works is it's beautiful. It's so simple. The mother puts two key proteins at opposite ends of the egg. Where there's a really high concentration of a protein called becoid, the message is basically screaming, build the head right here. At the other end, a high concentration of nanos protein signals, okay, this is the tail end. The cells that form in the middle, they just read the chemical gradient between those 2 points. It's an incredibly elegant system for setting up that main head-to-tail axis. But, nature loves to have options. So not every animal does it that way. Take frogs, for example. Their whole approach is different. The blueprint isn't preloaded. It gets drawn the very instant that fertilization happens. The exact point where the sperm enters the egg kicks off this massive internal reorganization.
And this shakeup creates this really special area called the gray crescent. Now, this little patch of cells becomes what scientists famously call the organizer. You can think of it like a master construction foreman on a job site. It starts issuing orders, directing all the other cells around it, and telling them how to build the back, the spinal cord, the entire nervous system. So that one single event, sperm entry, sets the whole body plan in motion. And figuring out how this organizer works, that's a massive goal in regenerative medicine. The dream is to one day use these principles to tell our own cells to build new tissues on command.
Okay, so our blueprint is drawn. We know where the head is, we know where the tail is. But how do you get from that basic grid to something as ridiculously intricate as an eye or a hand? Well, that brings us to the next phase, building the actual structures. So here's the secret. Cells don't just sit there and passively read their coordinates. No, they are constantly talking to their neighbors. And this process, this cellular conversation, is called inductive signaling. It's where one group of cells sends out a chemical signal that basically instructs the cells next to it what they need to become.
The way our eyes develop is the perfect example of this. So first, a little piece of the developing brain called the optic vesicle actually reaches out and physically touches the outer layer of skin. That touch is the signal. In response, the skin cells that got touched start to thicken up, and then they fold inward, and they eventually become the lens of the eye. The brain literally tells the skin, hey you, right here, you're going to become a lens. It's this incredible coordinated dance, and you can see how a tiny misstep in that conversation could lead to congenital eye defects.
And this very same idea of coordinated signaling is what builds our arms and legs. It's not just one signal either. To build something like a hand, you need at least 2 different key signaling centers that have to work together in perfect harmony.
Let's break those down. First, you have the AER. It's at the very tip of the little limb bud, and it controls the outward growth. It's the signal that says, keep growing longer, making sure your arm goes all the way from your shoulder to your fingertips. At the same time, you have the ZPA, which is on just one side of that limb bud. This controls the front to back pattern. It's the signal that tells the body where to put your thumb versus where to put your pinky. The precision here is just astonishing, and when these signals get disrupted, it can cause limb malformations, which really drives home how critical this cellular teamwork is.
All right, so we've got a body plan. We're building organs, but none of that is any good if all the parts aren't connected. The next monumental challenge is wiring the whole thing up with a nervous system so the brain can talk to the muscles and your skin can talk back to your brain.
So how does this happen? Well, each individual neuron sends out what you can only describe as a tiny, fearless explorer. It's called a growth cone, and its job is to navigate through the incredibly dense and complex terrain of the developing embryo to find its one specific target, which could be centimeters away, a huge distance on a cellular scale.
So how does this little explorer know where it's going? It's not just wandering around randomly, it's actually following a map made of chemical road signs. The whole guidance system works through something called chemoattraction and repulsion, which literally means the growth cone is pulled toward come here signals and pushed away from go away signals.
To guide all these explorers, the embryo lays down these chemical trails. Proteins like netrins act like a trail of breadcrumbs, attracting the growth cone and telling it, this way, come this way. At the same time, other proteins, like SLIT, act like giant repellent force fields or do not enter signs, making sure the neuron doesn't grow into the wrong neighborhood. It's this beautiful molecular game of follow the leader and red light, green light. And get this, scientists are studying these exact signals right now, hoping to one day create artificial trails to help guide nerve regrowth after a spinal cord injury.
Now, you would probably think that once an animal is born, the construction is pretty much done. But for a lot of species, birth is really just the end of Act 1. Sometimes there's a hugely dramatic second act where the entire body gets completely torn down and rebuilt.
And this, of course, is metamorphosis. And it's not just about getting bigger, it's a complete hormonally controlled reboot of the entire developmental program. You have a swimming, gill-breathing tadpole that literally dissolves its own tail and grows legs to become a frog that lives on land. A crawling caterpillar turns into a flying butterfly. It's a total teardown and rebuild.
In insects, this whole incredible transformation is run by a brilliant hormonal clock. There's a molting hormone called ecdysone. Every time it pulses, the insect molts. But what it molts into all depends on another hormone called juvenile hormone. As long as the levels of juvenile hormone are high, the larva just molts into a bigger larva. Simple. But when those levels start to drop, the next molt turns it into a pupa. And when the juvenile hormone is finally all gone, that last molt produces the adult. It's that simple of a switch.
And that brings us to one last fascinating twist. What if a really critical piece of the final design isn't written in the genetic blueprint at all? What if the environment gets the final say?
This is where things get truly wild, and it kind of turns everything we think we know on its head. For many reptiles, their sex isn't determined by X and Y chromosomes like it is for us. Nope, it's determined by the temperature of the nest where the egg incubates. For most turtles, cool nests make males and warm nests make females. But for alligators, it's the exact opposite. Warm nests make males, so the final critical decision of whether to build a male or a female body is made not by genes, but by the temperature of the sand. And this has huge real-world consequences right now, as climate change threatens to seriously mess with the sex ratios of these already vulnerable populations.
So from drawing the very first coordinates on a cellular map, to wiring up a brain, and even letting the weather make the final call, building an animal is a process of just breathtaking complexity and elegance. Which leaves us with one last big thought to chew on. Now that we're finally starting to really read and understand this amazing blueprint, what comes next? If we can read the architectural plans of life itself, can we one day learn to rewrite them?